Chromosomal theory and genetic linkage. Long before chromosomes were visualized under a microscope, the father of modern genetics, Gregor Mendel, began studying heredity in 1843. With the improvement of microscopic techniques during the late 1800s, cell biologists could stain and visualize subcellular structures with dyes and observe their actions during cell division and meiosis. With each meiotic division, chromosomes replicated, condensed from an amorphous, no constant shape, nuclear mass into distinct X-shaped bodies, pairs of identical sister chromatids, and migrated to separate cellular poles. Chromosomal theory of inheritance. The speculation that chromosomes might be the key to understanding heredity led several scientists to examine Mendel's publications and re-evaluate his model in terms of the behavior of chromosomes during mitosis and meiosis. In 1902, Theodor Boveri observed that proper embryonic development of sea urchins does not occur unless chromosomes are present. That same year, Walter Sutton observed the separation of chromosomes into daughter cells during meiosis. Figure 1. Together, these observations led to the development of the chromosomal theory of inheritance, which identified chromosomes as the genetic material responsible for Mendelian inheritance. The chromosomal theory of inheritance was consistent with Mendel's laws and was supported by the following observations. During meiosis, homologous chromosome pairs migrate as discrete structures that are independent of other chromosome pairs. The sorting of chromosomes from each homologous pair into pregametes appears to be random. Each parent synthesizes gametes that contain only half of their chromosomal complement. Even though male and female gametes, sperm and egg, differ in size and morphology, they have the same number of chromosomes, suggesting equal genetic contributions from each parent. The gametic chromosomes combine during fertilization to produce offspring with the same chromosome number as their parents. Despite compelling correlations between the behavior of chromosomes during meiosis and Mendel's abstract laws, the chromosomal theory of inheritance was proposed long before there was any direct evidence that traits were carried on chromosomes. Critics pointed out that individuals had far more independently segregating traits than they had chromosomes. It was only after several years of carrying out crosses with the fruit fly, Drosophila melanogaster, that Thomas Hunt Morgan provided experimental evidence to support the chromosomal theory of inheritance, genetic linkage and distances. Mendel's work suggested that traits are inherited independently of each other. Morgan identified a one-to-one correspondence between a segregating trait and the X chromosome, suggesting that the random segregation of chromosomes was the physical basis of Mendel's model. This also demonstrated that linked genes disrupt Mendel's predicted outcomes. The fact that each chromosome can carry many linked genes explains how individuals can have many more traits than they have chromosomes. However, observations by researchers in Morgan's laboratory suggested that alleles positioned on the same chromosome were not always inherited together. During meiosis, linked genes somehow became unlinked. Homologous recombination. In 1909, Franz Janssen observed chiasmata, the point at which chromatids are in contact with each other and may exchange segments prior to the first division of meiosis. He suggested that alleles become unlinked and chromosomes physically exchange segments. As chromosomes condensed and paired with their homologs, they appeared to interact at distinct points. Janssen suggested that these points corresponded to regions in which chromosome segments were exchanged. It is now known that the pairing and interaction between homologous chromosomes, known as synapsis, does more than simply organize the homologs for migration to separate daughter cells. When synapsed, Homologous chromosomes undergo reciprocal physical exchanges at their arms in a process called homologous recombination, or more simply, crossing over. To better understand the type of experimental results that researchers were obtaining at this time, consider a heterozygous individual that inherited dominant maternal alleles for two genes on the same chromosome, such as AB, and two recessive paternal alleles for those same genes, such as AB. If the genes are linked, one would expect this individual to produce gametes that are either AB or AB, with a 1-to-1 ratio. If the genes are unlinked, the individual should produce AB, A-flat, AB, and AB gametes with equal frequencies. According to the Mendelian concept of independent assortment, because they correspond to new allele combinations, the genotypes A-flat and AB are nonparental types that result from homologous recombination during meiosis. Parental types are progeny that exhibit the same allelic combination as their parents. Morgan and his colleagues, however, found that when such heterozygous individuals were test-crossed to a homozygous recessive parent, AABB times AABB, both parental and nonparental cases occurred. For example, 950 offspring might be recovered that were either AABB or AABB. But 50 offspring would also be obtained that were either AB or AABB. These results suggested that linkage occurred most often, but a significant minority of offspring were the products of recombination. Genetic maps. Janssen did not have the technology to demonstrate crossing over, so it remained an abstract idea that was not widely accepted. Scientists thought chiasmata were a variation on synapsis and could not understand how chromosomes could break and rejoin. Yet, the data were clear that linkage did not always occur. Ultimately, it took a young undergraduate student and an all-nighter to mathematically elucidate the problem of linkage and recombination. In 1913, Alfred Sturtevant, a student in Morgan's laboratory, gathered results from researchers in the laboratory and took them home one night to mull them over. By the next morning, he had created the first chromosome map, a linear representation of gene order and relative distance on a chromosome. Figure 3. As shown in Figure 3, by using recombination frequency to predict genetic distance, the relative order of genes on chromosome 2 could be inferred. The values shown represent map distances in centimorgans, cm, which correspond to recombination frequencies in percent. Therefore, the genes for body color and wing size were 65.5 minus 48.5 equals 17 cm apart, indicating that the maternal and paternal alleles for these genes recombine in 17% of offspring. On average, to construct a chromosome map, Sturtevant assumed that genes were ordered serially on thread-like chromosomes. He also assumed that the incidence of recombination between two homologous chromosomes could occur with equal likelihood anywhere along the length of the chromosome. Operating under these assumptions, Sturtevant postulated that alleles that were far apart on a chromosome were more likely to dissociate during meiosis simply because there was a larger region over which recombination could occur. Conversely, alleles that were close to each other on the chromosome were likely to be inherited together. The average number of crossovers between two alleles, that is, their recombination frequency, correlated with their genetic distance from each other relative to the locations of other genes on that chromosome. Considering the example cross between AABB and AABB above, the frequency of recombination could be calculated as 51 thousandths equals 0.05. That is, the likelihood of a crossover between genes A and B was 0.05, or 5%. Such a result would indicate that the genes were definitively linked. but that they were far enough apart for crossovers to occasionally occur. Sturtevant divided his genetic map into map units, or centimorgans, cm, single quote, in which a recombination frequency of 0.01 corresponds to 1 cm. By representing alleles in a linear map, Sturtevant suggested that genes can range from being perfectly linked, recombination frequency equals 0, to being perfectly unlinked, recombination frequency equals 0.5. When genes are on different chromosomes or genes are separated very far apart on the same chromosome, perfectly unlinked genes correspond to the frequencies predicted by Mendel to assort independently. In a dihybrid cross, a recombination frequency of 0.5 indicates that 50% of offspring are recombinants and the other 50% are parental types. That is, every type of allele combination is represented with equal frequency. This representation allowed Sturtevant to additively calculate distances between several genes on the same chromosome. However, as the genetic distances approached 0.50, his predictions became less accurate because it was not clear whether the genes were very far apart, on the same chromosome or on different chromosomes. In 1931, Barbara McClintock and Harriet Creighton demonstrated the crossover of homologous chromosomes in corn plants. Weeks later, homologous recombination in Drosophila was demonstrated microscopically by Kurt Stern. Stern observed several X-linked phenotypes that were associated with a structurally unusual and dissimilar X chromosome pair in which one X was missing a small terminal segment, and the other X was fused to a piece of the Y chromosome. By crossing flies, observing their offspring, and then visualizing the offspring's chromosomes, Stern demonstrated that every time the offspring allele combination deviated from either of the parental combinations, there was a corresponding exchange of an X chromosome segment. Using mutant flies with structurally distinct X chromosomes was the key to observing the products of recombination because DNA sequencing and other molecular tools were not yet available. It is now known that homologous chromosomes regularly exchange segments in meiosis by reciprocally breaking and rejoining their DNA at precise locations. Mendel's mapped traits. Homologous recombination is a common genetic process, yet Mendel never observed it. Had he investigated both linked and unlinked genes, it would have been much more difficult for him to create a unified model of his data on the basis of probabilistic calculations. Researchers who have since mapped the 7 traits investigated by Mendel onto the 7 chromosomes of the pea plant genome have confirmed that all of the genes he examined are either on separate chromosomes or are sufficiently far apart as to be statistically unlinked. Some have suggested that Mendel was enormously lucky to select only unlinked genes, whereas others question whether Mendel discarded any data suggesting linkage. In any case, Mendel consistently observed independent assortment because he examined genes that were effectively unlinked. Section summary. The chromosomal theory of inheritance, proposed by Sutton and Boveri, states that chromosomes are the vehicles of genetic heredity. Neither Mendelian genetics nor gene linkage is perfectly accurate. Instead, chromosome behavior involves segregation, independent assortment, and occasionally, linkage. Sturtevant devised a method to assess recombination frequency and infer the relative positions and distances of linked genes on a chromosome on the basis of the average number of crossovers in the intervening region between the genes. Sturtevant correctly presumed that genes are arranged in serial order on chromosomes and that recombination between homologs can occur anywhere on a chromosome with equal likelihood, whereas linkage causes alleles on the same chromosome to be inherited together. Homologous recombination biases alleles toward an inheritance pattern of independent assortment. Thank you for watching BookTube channel. Make sure to smash the like and subscribe buttons to receive notifications about new contents.